right, it is 8 o'clock. So I see that Catherine and Gerardo are with us. That's good. We will go ahead and we will start the webinar. So good morning, everyone, or good afternoon, or whatever time zone you're in. And thank you for joining us today. Today we're going to be talking about our new SpectraVue leaf spectrometer. And we're going to talk about its applications in commercial agriculture and the 3 novel uses for it in leafy greens, cut flowers, and berries. And so we had a kind of a sneak peek of the instrument earlier last month, and now this is the official launch for the SpectraVue, and we're really excited about that. So before we get started, we'll do some introductions. So our moderator today is Susie Truitt. Susie is our distributor manager here at CID Bioscience. She's been with the company for over 8 years now, and she's awesome. And she will be basically moderating the chat for us, as well as the Q&A. On that note, some light housekeeping stuff. For all questions that you might have, please utilize the Q&A function in Zoom. And so if you have a question regarding any sort of content about this webinar, so if you're asking a question about something we talked about, something about the instrument, a feature you might be confused about, anything like that that has to do with this webinar, please use that Q&A box. It's denoted there in that image. It's on the bottom right of your screen. And then if you have an issue with something like technical difficulty, say a lack of sound or lack of video, then please use that chat box for that. But otherwise, please refrain from using that chat function for any sort of questions you might have. And so Susie will be monitoring the chat box. She'll be monitoring that. If you do have technical difficulties, we'll address those. But other than that, please use the Q&A function so at the end we can be sure that we address your question before we end the webinar. A little bit about myself, in case you don't already know, my name is Galen. I am an application scientist at CID Bioscience. I have my bachelor's degree in biochemistry and a master's degree in food science from Michigan State University. I'm an IFT certified food scientist. In my previous roles, I have managed an analytical food laboratory and worked as a food safety consultant for the produce, food manufacturing, and cannabis industries. So today is going to be a little bit different than what we normally do and a little bit more exciting. So we just did the introductions, and then I'm going to jump right into talking about a new feature of this SpectraVue, which is building chemometric models. And then we're going to jump right from that into our guest speakers. So today we have Dr. Catherine Simpson and Dr. Gerardo Nunez, who are here to present their impressions of the SpectraVue and how they've been utilizing it in their research. And then from there, I will just do a product demonstration of the CI710S SpectraVue. And we'll do a walkthrough of all the different menus. I will take a couple of example scans, and then from there we'll go into the Q&A, and you can ask whatever questions you have of either myself, the instrument, or anything on Dr. Simpson's or Dr. Nunez's research. So without further ado, let's talk about this new feature of the SpectraVue. So in the previous launch webinar, if you attended, and I'll also be demonstrating it later today, but the kind of main function of this SpectraVue instrument is to utilize these built-in indices or customized indices to look at things like chlorophyll content or anthocyanins or carotenoids. But this instrument is also capable of so much more. So in this new product, we decided to be able to integrate the ability to create quantitative partial least squares regression models or any kind of chemometric models that you want using this spectroscopy. And so what I did for the purpose of this webinar and to help demonstrate how this works is I created a demo model for predicting moisture content in store-bought packaged organic baby spinach. And so how this works is essentially I took a bunch of spectral data by taking scans of baby spinach leaves, and then I took destructive measurements of those leaves by dehydrating them and getting moisture values from that. and I combined those two values, the spectra and the analytical result, and by compiling those together in our model builder software, I was able to get a chemometric model that predicted fairly well for moisture content in this packaged organic baby spinach. And so to walk you through that a little more in depth, the process for building a model, and this is kind of a loose outline of exactly what you would do for most other types of models you might want to build using this instrument. Initially, what I did is, went to the store, our local retailer, and I bought some packaged organic baby spinach, set up all the equipment that I needed for the analysis, made sure I had everything ready to go. And then I started by taking a calibration or performing a calibration of the 710S using our light and dark calibration standards. And those actually come with the instrument, and there we do have a nice video of that online now of how to actually perform the calibration. It takes, maybe 30 seconds at most to do this calibration, and it's a good idea to just do it every time before you start doing your data acquisition for the day. Or if you notice that spectra start to look really bad, that I have not encountered that yet in my extensive use of using this, but that's another situation in which you might want to calibrate. So I always calibrated it right before I started doing my data acquisition for the day. I never really had to recalibrate it again after that for the rest of the day. But then after I calibrated, what I did was I actually went and collected all that spectral data. And so the settings I used for this model building process is I used the automatic integration time on the instrument. I did 0 smoothing. So that means it didn't, I am only taking the spectral data at the specific wavelengths and not averaging any sort of, multiple wavelengths together. And then I did 16 scans to average, which essentially means instead of it taking a single scan, which, only takes, a millisecond or less, I actually did what it essentially does is it takes 16 scans in a row and it averages that value for that scan. It's just a way of ensuring, for me, ensuring that I'm getting the most accurate spectra I can from that single scan that I took. And so I did this in absorbance mode. So this is all absorbance-based model, and I did this I did 1 scan per baby spinach leaf. So if I was being even more robust about this, which I should, if I was intending on using this, pardon me, if I was intending on using this for, actual industry purposes, this model collection, everything would have been amplified a lot. So I would have taken more than 1 scan per leaf. I would have taken way more scans to use in this model. But this is just for demonstration purposes. So I did 1 scan per baby spinach leaf just to kind of show how the proof of concept. So from there, once I've taken the scans, immediately I took a scan of a leaf, and then I immediately went and I weighed that in order to determine the fresh weight. And once I had that fresh weight, because if you let it sit out too long, it's going to naturally dehydrate. So I wanted to make sure it reflected a fresh weight that was as close as possible to the fresh weight when I scanned it. So I did that in rapid succession. And then from there, once I did that process of taking a scan, getting the fresh weight for all the leaves, I then put them in the dehydrator. And so I dried them at 70 degrees C for 3.5 hours. And then after 3.5 hours, I removed a subsample of those leaves and I weighed those. And then I just put them right back into the dehydrator for another 30 minutes. And so after that 30 minutes, then I reweighed that same subsample, and I made sure that they were at constant weight. So for me, my criteria was if it was a less than 0.001 gram change, then I knew that those leaves were at a constant weight, meaning that they weren't going to lose any more moisture. And so once they met constant weight, I then removed them from the dehydrator, weighed all of the leaves in succession, and got from there the analytical values for moisture content. And so I exported all the data, all the spectra from the instrument. I used a wavelength range of 729 to 975 nanometers to build the model. And I used the corresponding analytical values for each of those samples, for each of those spectra. And I compiled it all using our model builder software. And it spit out for me a PLS model, which predicts fairly well moisture content in this baby spinach. And so then I did collect additional scans with the instrument. This is all on a single instrument, so I did collect additional scans of baby spinach leaves with the SpectraVue and did more destructive analysis for a validation set. And the predicted values from the SpectraVue were then compared to the analytical values. So in this next graph that I'm showing you, it is actually showing the validation set and how it looks with the predicted value being on the y-axis and the destructive value being on the x-axis. So it's kind of like saying, you know, the true or the correct value determined by wet chemical methods is on the x-axis, and what the instrument is predicting is on the y-axis. And so as you can see from this, we actually had pretty good prediction for a very small, you know, demo model. It actually did predict pretty well. And now part of that is attributed to the fact that this was all packaged baby spinach. It's all pretty much in the same moisture range, between 90 and 100. There was one leaf that I did see that, you know, that was kind of outside of our calibration or our test set range, but it still managed to predict fairly well. But other than that, it was a pretty good model with an R squared of 0.95, predicting fairly accurately. And you know, I see this as promising news for further model building for moisture and leafy greens. So, you know, this could be expanded to, you know, if I really wanted to make this robust, I would take multiple scans per leaf, like I said, and I would do it over multiple days with different batches, different brands of spinach, all sorts of different factors. Ideally, doing it with spinach all the way from, if you really wanted the most robust model, all the way from the farm, you know, taking measurements of spinach while it's still not even harvested yet, all the way through the supply chain to when it's packaged. And then from there, that model would, you know, for a fact at any stage in the supply chain, you could predict moisture content in that baby spinach. So, and this can be applied across all the different types of leafy greens. So if you're a commercial producer of any sort of leafy greens, looking for a rapid solution to do some sort of quality assessment, looking at moisture content, looking at, you know, chlorophyll or anthocyanins or some other any kind of analyte like that, micronutrients, then this is an instrument that I think you would be highly interested in. So that was the model building process, and I'm actually currently working on a new model now, looking at shelf life for baby spinach, which is actually going to be a more robust model. So be on the lookout in the future. Once I am done with that, I'll be making an application note for that. So that's going to be an exciting model as well to see how well the SpectraVue is able to predict shelf life for the baby spinach. So that is how we build models with the new SpectraVue, which is an exciting new feature. And what we're going to do now is jump right into some of our researchers that are outside of our facility, that are independent from us, ask them how, you know, see how they've been using our instrument, using the SpectraVue, and how they've been liking it. So first up, we have Catherine Simpson, who is an assistant professor in the Department of Plant and Soil Sciences not at Texas A&M anymore, but at Texas Tech University. And she actually was a student at Texas A&M for a very long time, doing a lot of research there, and doing stuff with citrus, looking at abiotic stress physiology. And with her postdoc, she was looking at tree physiology and insect populations. And then she actually started doing her research, or started as an assistant professor at Texas A&M. Now she's at Texas Tech and using the SpectraVue as one of our beta testers. And so Catherine, if you want to go ahead, I'll stop sharing my screen if you want to go ahead and load up your presentation, and we'll see how she's been using it.
Okay, thank you. Um, Give me 1 second. I think that should be it. Can you see it?
Yep, I see it right there. Thank you.
Okay, so thank you. My name is Catherine Simpson. I'm an assistant professor of urban horticulture and sustainability here at Texas Tech University. And my research, I actually just started here last year, so we don't have a lot of research projects going right now, right? But currently we're working with edible flowers and vegetable crops, especially in greenhouse and controlled environment systems. So one of the things we wanted to do is just see what pigment distribution and different other bioactive compound distribution in different types of plants is. And we've been using the SpectraVue for that. Some of the things, if I can get it to move forward. Okay, here we go. Um, Pigments, as we know, have many functions in plants. Anthocyanins are antioxidants, chlorophylls drive photosynthesis, and they can also affect photosensory pathways and photosignaling within the plants, and occasionally offer photoprotection depending on the type of pigments. So this is one of my students, Avishkar, and he's doing research on violas. And we have two experiments that we use the SpectraVue on. Experiment 1 was on viola leaves. We used them on the viola tricolor, Johnny jump up plants in different modified substrates. So we had different rates of biochar with the soilless media. And previous studies have shown significant differences in plant total polyphenol concentrations, as well as other chemical components within the plants. So we wanted to see what was reflected in the SpectraVue in these very young plants too, because we just potted them probably 2 or 3 weeks ago whenever I first took the first sample with the SpectraVue, and I took another one, but I didn't have time to put the results on here. And in experiment number 2, I just wanted to see what the pigment distribution in petunia flowers was. We had 4 colors of petunias, and they're vastly different colors. We had hot pink, light pink, white with purple, and light purple. So you're going to see the different pigments and how they are represented by the SpectraVue here. With the viola leaves, pigments were not significantly affected by the substrate treatment yet. However, if you look here, we do see that there are differences. The control is gray, then we have 10% biochar, 25% biochar, and 50% biochar. The chlorophylls were fairly the same across each of these because they hadn't been in the substrate for very long. The anthocyanin and carotenoid distribution in the viola leaves was a lot higher in the control compared to the rest, but it wasn't significant, probably because there was a high variation at this time. However, I will say that I did take measurements again, and we're seeing a huge difference in not only the anthocyanins, but the flavonoids. And one of the biggest things we've seen is in the 50% biochar treatments, we've got, I want to say, probably 10 to 20 times as much flavonoids than compared to the control treatments. And the petunias, what we did see was there was a significant difference in anthocyanins and chlorophylls based on their petal color. We had higher anthocyanins in the hot pink compared to the other colored petal flowers. And with the chlorophyll distributions, it was pretty much inversely proportional. We had higher concentrations of the chlorophylls in the petals that did not have higher concentrations of anthocyanins. And if you wanted to see just a little bit about what colors they were and the pigment distribution within them, these are the chlorophylls and these are the anthocyanins compared to each of these. So we do see that it is different, even if, you know, visually we don't see a huge like physical difference. There are reflections within the spectra that, you know, even though it is pink, the anthocyanin concentration is a lot higher. It's a lot more intense, a lot more concentrated whenever the pigment is a little bit more vibrant. And so with that, I guess at the end, if you have any questions, I'll be happy to answer them.
Awesome. Thank you so much, Catherine. And then I will go ahead and share. If you could stop sharing your screen, I will go ahead and share my screen again.
There we go.
A little quick transition here. So thank you, Catherine, for that. And now we're going to talk to Gerardo Nunez, Dr. Gerardo Nunez. And he is, let's see if I can actually pull up the, there we go. So Dr. Nunez is a horticulturist and an educator, and he is from Ecuador, and he is now a professor. He got his PhD from the University of Florida, is now an assistant professor at the University of Florida Horticultural Sciences Department. He's mostly focused on abiotic and biotic stimuli and how they impact horticultural crop quality, productivity, and resilience. And he's focused mostly on berry crops. And so I think we'll hear from him today about some blueberries, if I'm not mistaken. So I will go ahead and stop sharing now. And Gerardo, if you want to go ahead and share your screen.
All right, well, hi, everybody. It's my pleasure to be here today to share a brief update of some of the work we have been doing using the SpectraVue. Today, I have the pleasure of talking a little bit more about an experiment that my student, Cecilia Ruber-Heller, has been conducting throughout the summer. And I'm happy to know that we get to share this really early preview into how we're using leaf spectral signatures to diagnose ammonium toxicity in young blueberry plants. So to start, let me give you a little bit of context for the reasons for our research. Ammonium is the preferred form of nitrogen that blueberries take up. And this is kind of an anomaly. A lot of other crops do not do very well with ammonium. And the reason for that is that they can run into issues of ammonium toxicity. Now, southern highbush blueberries thrive in acidic soils. And in those acidic soils, ammonium is actually the most available form of nitrogen for the plants. So these plants are very biologically adapted to taking up ammonium. The one exception we find are plants that are coming out of tissue culture propagation pipelines. And as tissue culture propagation is becoming more important for the blueberry industry, what we're seeing is that more often than not, these plants can exhibit ammonium toxicity issues. Now, ammonium is perfect for full-grown blueberries, but what we're trying to do is we're trying to figure out what the best way to grow those very young plants is. In that context, Cecilia and I set up an experiment where we hypothesized that we could use leaf spectral signatures to track the onset and the severity of ammonium toxicity in tissue culture derived microcuttings of blueberries. We used a southern highbush blueberry cultivar from the University of Florida called Snow Chaser. And we grew these plants in a rhizobox system that allows us to take a look at the above ground and below ground part of the plant. A look at the system itself is there on the right. We fertigated these plants with a nutrient solution that we know would be optimal for full-grown blueberries, which would be a solution with 100 ppm of nitrogen, and nitrogen being delivered from ammonium sulfate. As we expected, we were going to see ammonium toxicity issues in these plants. When we tracked growth both above ground and below ground for those plants, we saw that there was a critical period where those plants were exhibiting ammonium toxicity issues, and that was during weeks 3 to 5. If you noticed, in panel A of the graph, there's not a whole lot of growth above ground happening between weeks 3 and 5. And that was also the time period where we saw the development of ammonium toxicity symptoms. Ammonium toxicity in the case of these blueberries was not evenly spread throughout the cane. It was actually affecting more the older leaves. And what we did is, what we thought was that this was the perfect opportunity to start using the SpectraVue to see what we could find out about those leaves and about the onset and the severity of ammonium deficiency using leaf spectral signatures. So to start, we went around and measured for 40 plants, one measurement per plant. We collected spectra from a asymptomatic leaf, which is always going to show as blue colors or blue diagrams in my presentation, and symptomatic leaves, which is always going to show orange. To start, we went ahead and tracked some of the commonly used absorbance-based measurements like normalized difference vegetation index, so the NDVI, also the chlorophyll NDVI, total chlorophyll as measured with the indexes that are built into the sensor, and of course the classically used SPAD. What we found was that these absorbance-based indices were able to distinguish very clearly between symptomatic and asymptomatic leaves. And this was terrific news for us because this was the first indication that indeed we were able to use the sensor to track the onset of this particular form of stress for the plants. Then we went forward, and this is the step that we're currently on. We are now taking a look not at the indices, but in fact, we want to take a look at the entire spectrum. And we're trying to identify signatures in this spectrum that are related to ammonium excess and potassium deficit. We know that those come hand in hand, so we're trying to relate our findings at the spectral level with biochemical findings in those leaves, so that we're able to further develop our understanding of how this form of stress affects young blueberries and how we can use the SpectraVue to be able to assess ammonium stress readily and easily in young blueberry plants. So as I mentioned, this is a work in progress. I am still sifting through a lot of the data. The project is very, very data rich because that's the kind of data set that spectral research creates. If you have any questions or thoughts, please feel candid about reaching out, and I'll be glad to talk more about our methods and about what we're hoping to find out through this line of research.
Awesome. Thank you so much, Dardo. That's honestly super interesting. I can't wait to see what more comes out of that research, so you'll have to keep us updated on that. Okay, so thank you to both Dr. Simpson and Dr. Nunez for presenting on how they are utilizing their SpectraViews right now. And so what we'll do is we'll go ahead and I will start a live demo. Give me 1 second here to actually get this linked up so I can do a screen share with you guys and actually show you what is on my screen with the SpectraVue. So go ahead and share. Okay, so I'm using the SpectraVue right here. I'm in my house giving this webinar, and hopefully you guys can all see this home screen of the actual SpectraVue. So this is what the home screen looks like. You see there's a top half that has where the spectrograph is, the bottom half is where the indices will display, or the calculations. And so I'm just gonna go ahead and start from the bottom menus, and that gear is the settings menu. And so here's where you're gonna be able to adjust all the different types of settings. So all the general settings, spectrometer settings, how your spectrograph displays, what wavelengths it displays between, as I connect to Wi-Fi, how you do your diagnostics for troubleshooting, and also just information about the spectrometer itself, like the serial number and things like that. This is also, importantly, where you will do the calibration. And now I didn't bring my calibration standards with me, which I absolutely should have, but this is a really quick process, and it guides you right through it. As you can see, it says step 1, insert the standard with the white pad facing down, press continue. You'll hit continue. It'll then prompt you almost immediately to say, put in the standard with the black pad facing down, do that. And then it'll say, take all the standards out, press continue, and then it'll be done calibrating, and you'll be ready to go. Really quick process. I recommend doing it just right before you start doing any sort of measurement collection or data collection, and then you'll be good to go and I have not experienced any difficulties with having to recalibrate at any point throughout the day when I've been using this. So I don't anticipate that being an issue. So to go back to the home screen, you just click the house icon. Now I'm back at the home screen. And so here is where I will actually go and we'll select an index, and then we will, I'll take a measurement. You can see how all the data is output. So first what I'm going to do actually is change my mode. So I clicked the right and left facing arrows, and now it gives me options of reflectance, transmittance, and absorbance. And so I'm going to change it really quick to reflectance. So now if I click it again, I should be highlighted on reflectance. And so if I want to add an index, it'll prompt you on the bottom. If you have no indices loaded, it'll say, please take a new measurement to see calculations. So that means that there's no measurements loaded, and I need to go ahead and load a measurement if I want one. You can also take scans without having any sort of index loaded and just get the spectra if that's all you want. But I'm going to go ahead for the purposes of this. I'm going to go ahead and see there's already one loaded. I'm going to actually go ahead. I swiped on that. I'm going to delete it, and I'm going to add by clicking the plus button. I'm going to add, and then I'm going to click this drop-down menu here where it says template custom. If you click that, here's a list of all of the different indices that are already built in. So I'm going to go ahead and first choose anthocyanin reflectance index. Since I switched to reflectance mode, I want to use a reflectance index. And then also you'll see there's the name of it. The notes say that it ranges between 0 and 0.2. The common range is for green vegetation 0.001 to 0.1. This also, the note section is where we'll sometimes put references to papers where we actually got the index from, things like that. And then the equation here is where you'll actually see how this index is calculated. And so when you go to create a custom index, you can actually update the equation yourself. If you have a new index that you want to test out for some sort of analyte, or if it's a nitrogen index or something like that, you can put that equation directly in here and create an index. And then it'll automatically calculate that for you when you go to take a scan. The bar color and the title color are just for customizing how it looks on the home screen. So the bar color is the color of the actual kind of background, I guess, of the name of the index when you go to the home screen. So that right now is purple. We'll keep it at purple. And the text, I'm sure the title color is the color of the text, which is black. So that's fine. And then to save it, I just click the save button, and now it's saved. And I'm going to go ahead and add one more just for fun. I'm going to go ahead and I'll add NDVI. And that one, the automatic color right now is light orange, but I want to make it green. So I'm going to make it green. And I'm going to save that one. Okay, so now I have these two calculations loaded. And what I'm going to do is I'm going to go back to the home screen by clicking home. And then I'm going to take a measurement of a leaf really quick. So I have a leaf here, and I'm going to go ahead, and I have not calibrated this instrument. So keep that in mind. These are not to be taken as super accurate numbers, but there we have it. That is the reading of this healthy-ish leaf that as healthy as it can be for being plucked off of a plant. And then I have another leaf here that's mostly dead, and I'm just going to do that one too, just to show you the comparison between the two values. So there we have it. That kind of makes sense to me. So looking at it, our first measurement of the healthy leaf is represented by the orange bar and the orange line in the spectrograph, and then our second measurement is the green line, or the green bar in the indices below. And so, as you can see, anthocyanins are higher in the dead leaf, the green line, the green bar, and NDVI is drastically lower because it is a mostly brown or almost black leaf. And so, with the healthy leaf, the NDVI was around 0.68, and that's a relatively healthy leaf here. And so that kind of makes sense to me. And so this is how you can have it display. You can always adjust by clicking the middle bar, this divider. You can always adjust exactly how much of your display is the spectrograph versus how much is the index. And if you want to go back to 50/50 screen, you click this black and white square on the right side, and your screen automatically goes back to 50% spectrograph, 50% indices. And so if you want to go and export these measurements now, what I can do is click the ruler, which shows the measurements. And from there, I can click a couple of options here. I can select all of these or deselect all by clicking this square, looks like kind of like a window. I can also tag certain measurements, like selected. If I select these measurements, which the only two that are selected right now have the colored bars next to them. If I want to tag them with a certain name, I can do that. If these are all from a certain plant, or if they're all from a certain group subset within my sample, then I would tag them with something. And then you can always filter your measurements by tag. You can delete them, and then you can also export them to a USB drive that plugs directly into the instrument here, or you can export it to a network drive as well. And so if you collected a bunch of spectra and then you only got the spectra, you forgot to load, say you forgot to load an index, right? What we can do then is actually go back and let's see if this is, oh, looks like I might be frozen now. Well, it looks like my internet can't really handle this connection right now, but essentially you can go back and you can always add a new index, and that'll automatically back calculate for you. So luckily I got through all the menus I actually wanted to show you before this kind of froze up. So yeah, that's the instrument and how it works. And we are really excited to be able to launch this product now. And I'll get back to, I'm going to stop sharing this screen. I'm going to go back to the...
Galen?
Yeah.
Real quick, just can you hold the SpectraVue up again and show them where the leaf actually goes in the leaf clip? Because I saw you with the leaf, but there we go.
All right, so I guess I can turn it around too if you really want to see. So here's the front of the screen, but I'm going to do it so I can see what I'm doing here. So that right there is the actual lens. And there's actually a lamp in the top and the bottom of this clamp. So that's what allows it to change between absorbance, transmittance, and reflectance. And so I just set the leaf right in there in the clamp itself. Boom. You can do it. And then, so if you have a large leaf like this, like what I was saying with the spinach when I was building the model, is that I only did one scan. Ideally, you could do multiple scans of the same leaf because you could do it at multiple different locations and move the leaf around. So that's how, and it stays in there, it clamps nice and tight, so you don't have to have a hand on it or anything. So just like that. Alrighty, so, I think I just accidentally did not share. Let me share my screen. And, all right, so that concludes our webinar today on the SpectraVue leaf spectrometer. So just some things in summation. So beyond the dozens of built-in indices that are commonly used across plant physiology, horticulture, agronomy, we also have this ability now to create customizable indices as well as these chemometric models that can predict analytes like moisture content, that can predict things like micronutrient contents. So it's a really cool, robust instrument. I'm really excited about it. And a lot of people can benefit from it. That's what I'm most excited to see, is all the different applications that people come up with. So obviously academic researchers are able to use this instrument, but I also see a lot of application potential in the agricultural industry. So people that are looking at blueberries or cut flowers or leafy greens that are in the commercial sector, you know, are going to be able to benefit from, you know, Dr. Simpson and Dr. Nunez's research, as well as the use of this instrument in the future. And leafy greens producers, as I mentioned, you know, being able to just look at quality traits within your supply chain rapidly and non-destructively is something that I think is a really invaluable tool. And so the SpectraVue, as I mentioned in the demonstration, As you saw, acquires this data, the spectra rapidly, non-destructively. All the data is transparent. When you export that data, it has all the information. It has the calculations for the indices you chose. It has the raw spectra data, the calibrated spectral data. So really all of it is transparent and in your hands, so you can do with it whatever you want to. And if you want more information about whether the SpectraVue is going to be something that can be applied in your situation, in your research, or in your sector of commercial agriculture, or in whatever your application might be, you can feel free always to contact myself or our other application scientist, Eric Munoz Garcia, and we're more than happy to discuss, whether or not this instrument could benefit you or not. If you want to reach out and get connected, we are on social media. You can follow us on Twitter, on Facebook, LinkedIn, as well as one of the best places to always keep in contact with us and to see what we're doing is to visit our website at www.cid-inc.com. Because of the pandemic, typically I would welcome you all to stop by anytime at our facility if you happen to be in Portland, Oregon, or the Washington, southern Washington area. However, please do not do that during this time. Instead, feel free to reach out for any sort of virtual, video calls or anything like that. You can always reach out to our sales department, sales@cid-inc.com. You can give us a call. And yeah, it was really great to be able to present this information to you guys. And now we're going to move on to our question and answer session. If you want to request a quote, feel free to click this link. I know that you can't actually physically click it on this presentation, but Susie will post it in the chat. So if you do want to request a quote to see what the pricing's like, if you do have any pricing questions or pricing related inquiries, please just request a quote. It's free, there's no obligation or anything to buy. That'll give you exact pricing for your area. And so, yeah, without further ado, let's go ahead and open up the question and answers, and we will address those one at a time. And so it looks like we have a good amount of questions here. And Gerardo and Catherine, if you guys also want to open up if you guys can open up your question and answers just in case there's questions for you guys. I will go ahead, I'll still read them out loud though. So our first question from Olga is, can the system be used to measure secondary metabolites such as alkaloids and terpenoids? So the short answer, I would say that's highly unlikely. That those terpenoids specifically, I don't think are going to be within the wavelength range of this specific spectrometer. So I think that you should be on the lookout in the future. We have something in the works for you. But in the wavelength range of 380 to 1300-ish, you're not probably going to see nanometers. You're probably not going to be able to get a quantitative model for terpenoids or alkaloids. The next question is, how do you measure the pigments? So I don't know if this is this is probably for Catherine, I'm guessing. Part of your experimental design, is that what the question is asking, I'm guessing? Like, how did you quantitatively measure your pigments?
Yes, we've got different methods to actually physically analyze destructed samples. In the lab, we use a Spectromax ID3 microplate spectrophotometer. There are different methods for different pigments. Chlorophylls, usually we use the Lichtenthaler method. Carotenoids as well. There's another method for anthocyanins and our total polyphenols and our total flavonols too. So each one of those has a different method. We're still working on adjusting it for the microplate reader, but most of ours, especially the total polyphenols, are good right now. Our anthocyanins, because they are very sensitive to heat and other things, we're still working on those methods. So it's been a little bit more challenging than we had wanted it to be to adjust it for those. But so far we're getting better. We're this close to getting that one done, but our flavonoid method is also complete.
Awesome. Thank you for that.
Mhm.
And so the next question is it possible to detect heavy metal ion presence in the leaf samples? So there is research out there that, and I think I actually presented on it in the previous webinar, but there is research out there that indicates that not necessarily heavy metal ions, but you know, nutrient ions like potassium, nitrogen, phosphorus are all easily detectable using spectroscopy. Heavy metals, because they're going to be so trace, I assume, unless I don't really know of situations where they would be in you know, in really high quantities. But I do recall that paper looking at I think they looked at copper as well, which I think they were able to get a model for. But I would say in general, where spectroscopy has issues for the most part is the really, really trace level. So things that still require HPLC, MS, you know, that kind of low level, you know, parts per trillion kind of detection levels. That is where spectroscopy still doesn't quite have that, you know, the ability to see levels that low. So I think it depends on the levels of your heavy metals, but I would say most likely not if they're in really trace levels. The next question is for Dr. Nunez, and this question asks, it's a very interesting project. Why did you use the rhizobox while you used the leaves for most of your analysis, and how does the box work?
Well, thank you for the question. We like to study nutritional issues and nutritional deficiencies by having simultaneous looks at roots and leaves. I think one of the older paradigms of plant nutrition was that we were very focused just on one organ. And part of the way we have been studying plant nutrition for some time is always keeping root shape, growth, and functionality in mind as we study the overall outcomes. Our intention with using the rhizoboxes is nothing other than being able to see those roots as they develop, as they colonize different parts of the soil, and in as much as we can quantitate that, we're able to get things like growth rates and root densities per square centimeter or square inch of soil.
Awesome. Thank you for that, Dr. Nunez. And that was a great question. And so moving on to the next question is scientists have reproduced the epidermal cells of rose petals that have particularly good anti-reflection properties and integrated the transparent replicas into organic solar cells. This resulted in a relative efficiency gain of 12%. Can SpectraVue measure reflectance for this application? Wow, that is a really cool application. Okay, so organophotovoltaics. So if we talk about actually measuring a solar cell itself, absolutely, you can measure reflectance of whatever can fit in this leaf clamp here. So you can look at the change in reflectance from a solar cell that is a typical organic solar cell versus a solar cell that has this rose petal epidermal cells transplanted into it, and see what the change in reflectance is based on the spectra. You could create a model to actually potentially quantitate that. So I mean, as long as basically your limitation there is I don't know what the actual solar cell looks like. If you can get it to actually fit in the clamp here, in the actual leaf clamp, then potentially, yes, it could work in that application. The next question is, could this be used as indirect nitrogen measure on growing corn? Absolutely, yep. This so actually in the previous webinar, which I recommend you check out, there's a couple of really good research articles in that. Nitrogen is one of the components that were actually quantitatively measured, creating a model with our older CI 710 miniature leaf spectrometer. And so you can build quantitative models for nitrogen. And if you want to do indirect nitrogen measurement, there's lots of different indices out there that are correlated to nitrogen content that you can find in papers, and you can plug those the equation for those indices right into the actual instrument and have it calculate those indices for you if you want a more just, yeah, kind of indirect measurement method. The next question is, how do you clean the sensor? Okay, so this is a, I mean, if you can see it, it's not recessed or anything, so they're likely not to get it, it's not going to get too dirty, but just using a microfiber cloth in general, just to use that to do most of the cleaning of it. If you need to, if it gets really dirty, you need to do a thorough clean. I think just mild soap and water is fine. I don't know. I don't know for certain if any sort of solvents are you know, harmful to it or not. But I think that in general, if you use just like a organic compound cleaner, like in the lab we used to use something called Alkanox, and there's other, you know, cleaners that clean out organic tissues really well. Then you can use that, a mild solution of that with water to clean off if it gets really dirty. The next question is, can we measure absorbance, transmission, as well as reflectance all at one time? So that's a good question. The short answer is there is a way to do that, but to do that, I think it would take longer than you would like probably for a scan on a per scan basis. But there is a way that you can have all three at once. It'll basically just do a series of absorbance and then transmittance and then reflectance scans. But for the most part, I think for most applications, you could probably find a way to only use one or the other. But if that's something that you need to do, then yes, it's possible. And the next question is it possible to get non-destructive measurements? And absolutely. So it is a non-destructive instrument in its nature. And so the instrument itself is always non-destructive. However, if you go to build a model, a chemometric model, that does require you to do initially a series of scans followed by destructive measurements of those same samples that you scanned. But in general, if you just want to use indices that you found in publications or that you found or that are already built into the instrument, then it's absolutely non-destructive. The next question is, when sampling the leaf, is the expectation that the leaf should have the same moisture throughout the entire leaf? Does the SpectraVue track the different moisture levels and records the different test locations of the leaf? So short answer is no. Basically, it's bad experimental design on my part, but because it was only for the purpose of demonstration, I was kind of being, I guess, a little bit lazy about it. I just did one scan per leaf. Technically, what you should be doing is sampling multiple locations around the leaf, and then, you know, using the average from that of those spectra that you took, you know, say you took 6 scans of one leaf, use the average of those 6 scans, and compare that, you know, that spectra then correlates to the complete total leaf, you know, moisture content. Because I dried, you know, I only scanned one location on the leaf, but I dried the entire leaf. So technically, you know, this experiment that I did, it is assuming that the moisture is distributed, you know, homogeneously throughout the leaf, that the moisture content in the location that I scanned is representative of the entire leaf moisture content. And so luckily with the spinach, it turned out to be that way, because the model actually turned out pretty well. But for a lot of leaves, it's not going to be that way. So I would highly recommend, if you're going to build a model, if you want to build a robust model, you take multiple scans of the leaves. The next question is, how about the mineral content such as nitrogen? How is the result so far? As I mentioned earlier, there's a great paper that was published not too long ago. I presented it in the last webinar that actually used our older version, the CI710, to create quantitative models for mineral content such as nitrogen and potassium and phosphorus and so on. And they had good success. So I think that it's completely doable. I think that it just requires some labor on the user's end to actually create the model. Okay, the next question is, what is the maximum OD and absorbance that can be measured, and at what SNR? That's a really great question. I actually do not know the answer off the top of my head to that question. So Shrenik, if you want to actually email me that question directly, I can ask our engineers to provide that information to you, because I do not know off the top of my head. I'm sorry. The next question is, can the system be used to measure water stress or photosynthetic rate? So photosynthetic rate would be difficult for this. I don't know of any spectral methods to look at photosynthetic rate that I know of. There might be out there. I just don't personally know of any. But water stress, absolutely. You can look at things like the water band index, or you can create a actual, moisture content model, and along with a bunch of other myriad of other indices that can help indicate water stress. There's lots of publications out there that have used spectroscopy to help kind of predict, the onset of water stress, or look at drought conditions and things like that. So it absolutely can be used for those applications. The next question is, are there batteries that need to be changed? If so, how much do they cost? So yes, all of our instruments are powered by rechargeable lithium-ion batteries, and they are just the standard, I don't know what the actual name of them is, the standard size rechargeable batteries, lithium-ion batteries. And I'm not sure off the top of my head how much they cost.
I can tell you that the instrument comes with 2 sets of batteries and a charger.
Yeah. So you'll never really, unless you're planning on measuring for over 12 hours straight, I would say that you're going to be fine having the 2 sets of batteries, and you could just have the charge set, the spare set on you. If you really need, if you're going to be measuring, for long extended periods of time, and you're going to have to change your batteries, then just carry the second charge set with you. Replace them when you need to, and then you can recharge them, both sets overnight, and then you'll have that, the ability to measure for up to, or for 8 hours straight again the next day if you need to. The next question is, how about the impact of fungus to the leaf quality? Is there a relationship to the spectral signature so far? So I feel like I've come across some articles that have investigated things like, disease and other, biotic stressors to plant quality and health. So you'll definitely will see changes in spectral signature with a plant that's highly infected with some kind of fungus versus a plant that isn't. But the exact nature of that relationship, I don't know. It's definitely dependent on the type of fungus, the type of plant. But I definitely think that that's something that you definitely could use spectra or spectroscopy in order to investigate. The next question is, are there variations of the SpectraVue for different sized leaves like Arabidopsis systems with really small leaves? So unfortunately, no. The answer to that question is no. This is the size of the SpectraVue, and this is the size of the aperture. So with things like grass type leaves or Arabidopsis type leaves, what you'll want to do is just actually measure multiple at one time if you can. In order to cover up as much of that aperture as possible without too much overlap, So you still want to try to get it to be a single layer, but you want it to cover as much of that aperture as you can so that you're not just measuring a bunch of empty negative space. So it can work, it just requires some adaptation on the experimental design end of things, I think, is the biggest challenge, I think, to overcome there. The next question is macro and micronutrient measurements, correlation to tissue testing or sap sampling. So I'm not 100% certain what this question is asking. Tom, if you want to go ahead and send me an email and kind of expand a little bit on what you specifically would like to know about with the tissue testing or sap sampling. But for the most part, if you're just asking if we can measure macro and micronutrient contents using spectroscopy, using the SpectraVue, the answer is yes. As long as, you know, some micronutrients that are in really, really low concentrations, like as I mentioned earlier, spectroscopy has some issues with things that are in very trace levels. So if it's a micronutrient that's at a very trace level, then likely not, but most of the macro micronutrients in plants are able to be measured using spectroscopy. And the SpectraVue will certainly work for that. The next question, I feel like I just answered, but the next question is asking, you can measure micronutrient content. Yes, like I said, yep, absolutely. As long as it is at something above trace levels, then the SpectraVue will be able to see that. The next question is, has anybody tried to identify citrus greening disease using leaf spectra with this machine? You know, I don't think so, but we actually, weren't we talking about citrus greening with the root scanner, I believe, with our root imaging system. But anyways, I don't know if anyone has actually tried using this for citrus greening disease yet, or at least tried using spectroscopy in general. But that would be a really interesting application, and I think something that definitely, you know, could be, the SpectraVue could absolutely be used. I think that when we were talking about citrus greening disease, they were talking about, we were talking about using the root scanner because they were trying to look at mitigation strategies for detecting it even before it displays anywhere on the actual plant itself. So looking at detecting that, even, you know, having the roots show signs prior to anything above ground showing signs. But still absolutely could work. The SpectraVue could work for looking to identify that as well.
Can I add something too? Because
yeah,
I did work in citrus previously. There are people that are using like NDVI and other spectral scanning things, like using drones to try to identify citrus greening disease in grove-wide care. So that could be some of the applications. Now for spectrophotometric analysis of the leaves, I don't know.
yeah. Yeah, all right, so thank you for that, Catherine. I appreciate the added input from somebody who actually has worked in the citrus research field. So the next question is for shelf life prediction of baby spinach, what indicators am I looking at? I can imagine that one of them is water content. So actually what I'm doing is I'm technically replicating another study that has already kind of done this with spinach, actually, specifically. And so, Michaela, if you want to go ahead and email me, I can send you that study. It'll be easier that way than me trying to, I think, get it to Susie to get it to you or some other kind of mediary system. So go ahead and just email me directly, and I will email you the paper that I'm basically replicating, essentially, at this point. But for right now, it's actually just going to be determining kind of age of the spinach. But yes, moisture content is another factor that is going to play into the shelf life of the spinach. The next question is, yes, but how is this instrument is useful in measuring? Yes, but how this instrument is useful in measuring pigments. So as you saw with Dr. Simpson's research, that there's a lot of built-in indices to look at things like anthocyanins, carotenoids, you know, chlorophyll. But for other pigments, I'm certain that there are other indices out there, or you can use the actual spectra and create, you know, your own predictive model based on destructive measurements of those specific pigments. If you're looking at other pigments besides kind of those the major ones. Yeah, so it's absolutely useful because this is a visible to NIR spectroscopy instrument. So this is absolutely capable of measuring pigments.
Yes, and it's non-destructive, plus you don't have to do the extractions. You don't have to have a lot of the chemicals and the equipment used for the destructive measurements and analysis.
Yep, thank you, Catherine. And so the next question is, what is the efficiency to measure the flavonols and plant nutrient correctly? So the indices aren't direct measurements of anything. Technically, really, they're not. They don't provide like a concentration value of grams per 100 gram or anything like that. They're just kind of a general indicator of concentration. However, if you go to build a chemometric model, then you can see what the actual, accuracy of that model is for predicting things like, or measuring things like flavonols or plant nutrients. As you saw in my validation graph, the R squared was 0.95. For the most part, the error there was less than 1% moisture for most of the predictions. And so, with a really robust model, you can get that accuracy that you need within, 1% of, moisture content, within, 1% plant nutrient content, things like that. It all really depends on how robust your model building process is and how robust your model is. The next question is, can the system measure THC, cannabidiol, other cannabinoids in marijuana plants? So likely not, unfortunately, with this wavelength range from 380 to 1300-ish. This system likely is not going to be able to quantitate those as strictly as you want. If it's strictly for research purposes and not for any sort of regulatory purposes, then it might be something that you might be able to do. I know that in the past when we've used this wavelength of spectrometer, we've been able to do total cannabinoids pretty well. But if you go to actually separate them out into all of their THC, THCA, CBD, all of those different actual individual compounds, that's when it gets to be more difficult, and it loses its accuracy. So I think that for spectroscopy, at least, you'll want to wait a little bit, and in the future we should absolutely have something for you. But with this specific spectrometer that's in the SpectraVue, unfortunately, I think only total cannabinoids is likely the only thing you'll be able to actually get accurately and quantitatively. The next question is, can we measure transmission with this instrument? Yep, you can. This has a transmittance, absorbance, and reflectance modes. The next question is there a module in the instrument to measure liquid samples? There is not, but be on the lookout. I would highly recommend you visiting our website because we might have something for you here relatively soon for the liquids. The next question is there any particular regression model required to measure a particular parameter in a quantitative way, or is it performed universally? So you can use whatever type. So this instrument, the actual only data output you get is all your calculations, the raw and the calibrated spectra. And with that, you can do whatever you want as far as chemometrics is concerned. You can build PCA models, you can build PLS models, you can build artificial neural network models, you can do whatever type of chemometrics you want, doesn't matter the actual methodology. And honestly, you might want to do it multiple ways and see which one performs better. So there is no universal way of actually creating the predictive models. It just requires patience and a lot of data, and all these different new types of methods for parsing through all this multifaceted data. The next question is it possible to detect the air pollutants which cause the damage? So if you want to look at things like pesticide residues or things like that, usually those are in such small concentrations that I really don't think you'll be able to create any sort of model to see that kind of levels. You still need the more advanced destructive technology to see pesticide residues or any sort of other pollutant residues like ash or smoke from all the wildfires that we've had or things like that. So I don't necessarily think, I can't say confidently that it would be able to see those kind of that low level of pollutants. The next question is, can it measure reflectance in cloudy, humid conditions with accuracy? It can measure reflectance in any conditions with accuracy because it's actually mostly just a closed system. So you're actually clamping down the leaf in between the lamp within here and the lamp within here. And so you're essentially creating this closed system that's not really exposed to the environment while you scan. So there should be no room for any sort of environmental factors to actually influence the reflectance measurement itself. The next question is a question that I should know the answer to, but I don't think I do off the top of my head. It is, what is the actual aperture size, the dimension of this aperture? So eyeballing it, I'm gonna say it's about a centimeter, a little less than a centimeter, or no, it's about a centimeter in diameter. I think about that. That's from eyeballing it, but you can always go to our website, and we have specifications on our website on the product page, so you can always go there to see what the actual specifications are for everything. The next question is, does the aperture have to be completely covered by the sample, or can part of it be uncovered? I do recommend you covering it as best as you can completely. And as I mentioned, if you are doing grass or Arabidopsis or really small leaves, you might want to, what you want to do is adjust your protocol so that you are measuring multiple leaves at the same time and covering up that lens as much as possible. The next question is it possible for stored leaves and dried leaves? Yeah, absolutely. You can use it, and so for any condition of leaf. And as I mentioned earlier with the solar cell question, you're not just limited. I mean, this is a leaf spectrometer, but really, if you can fit it and it fits over the aperture and it can fit within this clamp, it can be the spectra can be taken. So. Yeah, it will work for both stored and dry leaves. The next question is, can your measurement be affected by leaf temperature? The answer to that is yes. Spectra are influenced by temperature. So if you want to ensure that temperature isn't influencing your results, you will want to take measures in your experimental protocol to ensure that that doesn't happen. If you're building a model, you want to include all the different temperature ranges kind of evenly distributed throughout the model, throughout all your data. If you are just doing index readings, what you'll want to do is make sure that you're doing it either at this, at similar times of the day, or if you're doing it, in similar conditions. If you're out in the field, then for the most part, the reflectance measurements won't be influenced too much, but there is going to be some change. So you will want to take that into consideration if you're in the field. But if you're in a lab setting, or if you're in a commercial storage setting or anything like that, then you won't have to worry about the temperature fluctuation as much. The next question is, what is considered trace levels by the instrument for micronutrients in percent or ppm? Off the top of my head, I can't say for certain. The paper that in our last webinar, I presented that paper. I want to say, I don't want to say anything because I don't know if I'm going to say something that is inaccurate or not. So I would go back and watch our previous webinar, which was the pre-launch of the SpectraVue, and look at the papers that I presented, and see if they have more information on that. I don't know for a number for certain where it kind of drops off where we no longer can see, but I think that anything less than like 0.1% is going to start to be difficult to see for sure. The next question is, how does leaf pubescence affect the quality of measurement? I don't think it will affect the quality so the only things that are going to influence actual quality of measurement are things like if you don't have the entire aperture covered, or if you accidentally don't have the entire aperture covered in one measurement, and then in the next measurement you have it completely covered, or things like if you are measuring at wildly different, if your leaf is been in extreme heat conditions, and you take a measurement, and then you take a measurement of your next measurements you take in the middle of the night when it's really cold or something like that. So things that actually affect the quality of the spectra are usually those kind of physical factors but usually just you'll see a change in spectra based on leaf, the leaf pubescence, other conditions. But that change doesn't necessarily mean the quality of the spectra is worse. It just means that you're seeing an actual spectral difference because there is something different in the plant. All right, the next question is it suitable for testing leaves of different sizes and thickness? Yeah, so absolutely. It'll work for any, like I said, as long as it can cover the aperture, as long as it can fit in the clamp, then you should be fine. I think the one thing that people would probably find the most difficulty with is if you tried to measure like conifer needles. I think that'll probably give people the most difficult time. But other than that, I think that most plant tissues will be totally acceptable to use with the SpectraVue. The next question is, does it work with low light conditions? And the answer to that is yes, it'll work in any light conditions because, as I mentioned, it's a kind of a closed system. You clamp the leaf in, and then you get the lamp is built into the top of the clamp and the bottom. So you are essentially going to be able to take measurements. You can take measurements under any light conditions. The next question is how to calibrate the instrument to measure leaf chlorophyll content. Is it necessary for us to develop the calibration curve? So if you want to quantitatively measure chlorophyll content, if you don't want to use indices, all the different indices that have been developed to look at chlorophyll content, if you want to actually make a model to look at exact quantitative value of chlorophyll, then yes, the user will have to develop that calibration. All right, the next question is, let me take a drink of water because my throat's starting to get a little sore. The next question is, during measurement, how much would light intensity in the environment of measurement affect results variation? This is just like the dark light conditions, it will not affect the results. the actual spectra. All right, next question is, what is the current price? So go ahead, if you want pricing information, go ahead and fill out or click the link, the tiny URL, and request a quote, and we will have pricing information for you within 24 hours. And then the next question is, do we have a device that can be used to reduce the aperture size? So that's a good question. Unfortunately, we don't, and you know, we've attempted to do things like that with other instruments that we make, and we find that a lot of times it actually is more detrimental to when we do that, when we somehow manually or physically reduce that aperture size with some sort of adapter. Something about it makes it more difficult from the model building process, from what I've seen from other collaborators and customers' data. So I don't know if we will plan on doing that, but so the answer is unfortunately no. So for plants like tea, the chlorophyll content is not within the linear range of SPAD meter readings. Therefore, it is not possible to use SPAD meters for chlorophyll measurements. Would you think this instrument can be used to determine the high chlorophyll content from leaves like tea? Absolutely. So if you know that your chlorophyll measurements are going to be higher than what a the SPAD index or any other index can measure, then what you can do is create a model to actually quantitatively predict the chlorophyll content in your specific leaves. Load that onto the instrument, and then from there it'll just go. You'll be able to go and non-destructively predict. It will require some model building on your end, so you will have to be able to do destructive analysis of chlorophyll initially. But after you build the model, it'll all be non-destructive from there. So yeah, this instrument I think would definitely help in your situation. The next question is, can leaf spectroscopy predict leaf and forest traits? So yeah, lots of different leaf, parameters. I don't know, I guess, specifically what you mean by forest traits, but there's lots of different already built-in indices, lots of different things you can look at as far as detection of water stress or disease or macronutrient content, lots of different parameters for leaves that you can, which then can be extrapolated to whole plant levels. So I think that hopefully that helps answer your question. I'm not 100% certain what you're specifically asking about. The next question is, does the unit record temperature each time a sample is taken? Does it have an ATC function to normalize readings? So we actually don't record temperature. There's no temperature sensor within the actual, measurement aperture. So but we do have internal temperature measurements of the instrument itself. That's more on to make sure the instrument doesn't overheat kind of things. But for attenuating temperature kind of functions, I think that the best way to do that, to go about that, is to, if you are, I guess when you're building the model, like I said, if you're building a model, you'll want to incorporate those temperatures into the actual, data that you acquire, to make sure that everything, all the different variables within the temperature, all that variation is accounted for within your data set. Unfortunately, there is no, and this is something you encounter with all spectroscopy, there is no just like direct way to kind of correct for temperature. It varies with each instrument, and it varies. So there's no just kind of set formula that we can use to kind of correct for that. It varies with every commodity we measure, everything we do. It's very variable. So unfortunately, there is no like ATC function to normalize the readings for temperature. But I will actually, John, if you want to reach out to me, we can talk about some workarounds for your application. If you're concerned about temperature being a factor that introduces too much variation into your readings. So the next question is, from the measurement of leaf macro micronutrient content, is it necessary to have calibration curves for each plant species? Like for example, one for spinach, another for Swiss chard. For species, yes, I would recommend a different calibration curve, mostly because there's just too much physiological difference between those two specifically, that I wouldn't feel comfortable with a single model being able to predict accurately for both. But for things like, you know, different cultivars of the same, you know, fruit, or, you know, of the different cultivars of the same, I guess, species, would be a little bit more appropriate to do that if you want to incorporate multiple things into one model. So you want to do it with, as you know, things that are similar enough that you feel confident, but with two things that are so dissimilar, I would recommend two separate models. And then our last question here is it possible to predict key leaf traits associated with source sink balance and carbon nitrogen status with spectroscopy? So I actually think that one of the articles I presented on last webinar did talk about this. However, I think that, I mean, with nitrogen, absolutely. Nitrogen status, carbon nitrogen status, I don't know about actually looking at, you know, I don't know of any studies that have looked like over, you know, time to see, you know, the changes in the spectra, you know, associated with the source sink balance. But I think that would be something really interesting to see. And I think you will see differences. So I think that it is possible to, you know, after collecting that kind of data, to then build a model that might be able to predict you know, parameters that are associated with that. The next question is, what is the range of chlorophyll measurements which can be determined with this instrument? That is entirely up to you. So as I mentioned, the indices aren't any sort of direct correlation, or aren't any sort of direct chlorophyll measurement. They aren't direct anything. They're indirect measurements of these different pigments, or you know, of different you know, moisture or water, or other properties of the plants. So if you want to build a quantitative model, you would have to determine for yourself what the range is going to be based on the data set you collect for that model. So there is no, technically, if you're building a model, there's technically no limit, I guess, on what the range would be. All right, so that was 50 questions. Thank you so much, everyone, for being so attentive and for asking so many questions. Honestly, it's great to see that much participation. So that's the end of our webinar. And thank you again, Dr. Simpson and Dr. Nunez, if you're still if you're still on here. I know it's been a little bit longer than you probably anticipated, but I appreciate you staying around and presenting. And it was great to hear about your research. And I'm excited to see what more you guys do and how your research comes along. And again, thank you all for attending. If you want to request a quote, click that tiny URL link in the chat. I'll leave this up for a little bit longer. And then if you have any application questions, please reach out to me directly via email. Thank you, everyone.